Hello everyone. In this video series, I'm going to talk about antigen-antibody interactions. Okay, there are two examples of this type of interactions which are very common, like precipitation and agglutination. Okay, so in this video, we're going to talk about immune interactions or immune reactions, which have two types, like precipitation and agglutination. And in this video, I'm going to focus only on first type of antigen-antibody interaction, that is precipitation. Okay, it is the type of immune reaction. So this precipitation involves some type of immune complex or immune molecule in it, and these molecules are antigens and antibody. Okay, so in precipitation, antigens and antibodies are involved. Okay, both these antibodies and antigens are specific to one another. So this type of reactions have wide application in hematology. Okay, we can find out the concentration of antigen or antibody from the sample with the help of this precipitation technique. So the application of this technique is quantitative estimation of antigen and antibody. One can find out the unknown concentration of antigen or antibody from the sample with the help of precipitation technique. There are various examples of precipitation, like precipitation ring test, single radial immunodiffusion technique, double radial immunodiffusion technique, and immunoelectrophoresis technique. Okay, now we know that when any antigen come in contact with specific antibody, a non-covalent interaction develop in between these two molecules, antigen and antibody, and after binding of the antibody with the antigen, a small complex is produced, and such complex is called as immune complex. Okay, so these immune complexes are nothing but the antigen-antibody complexes. Antigen is a foreign substance that enter in the body, which may be bacteria, which may be virus, or they are proteins, while these antibodies are the protein which is made up by plasma cells, which is the subtype of B cell. Now, this antibody molecule get attached with antigen with the help of non-covalent interaction, which develop immune complexes. But initially, these complexes remain very small, so they always be soluble. Okay, they do not become visible in the solution. But we know that each antibody always have two antigen binding site. Okay, so the antibodies are always bivalent. If we consider the structure of the antibody, in that case, antibody molecule look like this, Y shape. Okay, the upper portion of this antibody is called as FAB, that is fragment antigen binding, while the lower portion is called as FC, that is fragment crystallizable. Okay, now this FAB portion have two antigen binding site. So each antibody can bind to two antigen at a time. So we call this antibody molecule as a bivalent structure. Okay, it has two valencies. Now, if antigen have two antibody binding site, in that case, such antigen also called as bivalent antigen. Okay, suppose this is the antibody which is specific for this antigen. Okay, and now the antibody binding site present on antigen is called as epitope. Okay. Suppose antibody is bivalent, an antigen which is specific for this antibody is also bivalent. That means this antigen have two epitope on its surface. Now in that case, this antigen with the epitope get attached to the antibody. Okay, and after binding, one epitope of the antigen still remain free, and this epitope can again bind to another antibody. Okay. So same sequence repeated. Each antigen bind with one epitope to one antibody and with another epitope to another antibody. That lead to development of cross linkage. Okay, so due to this bivalent and nature of the antigen and antibody, cross linkage occur, and due to this cross linkage, instead of the small immune complex, large complex is produced, because many antigen in a chain get attached to with the antibody and they cross link the antibody, and due to this large complex, this large immune complex now no longer remain soluble in the solution. It get precipitate out from the solution. Okay, and now this reaction is called as precipitation reaction. Okay, when any bivalent antibody get attached with the bivalent antigen, a large immune complex is produced. This large immune complex is sometimes called as lattice. And now this lattice get precipitate out from the solution, and this precipitation process or the reaction is called as this first type of antigen antibody interaction. Now remember that for precipitation to occur, antibody must be bivalent, and this antigen must be bivalent, or sometimes multivalent antigen is also lead to precipitation reaction. What is multivalent antigen? The antigen which have more than two epitope on its surface. Such kind of antigen also develop lattice, and this lattice ultimately give rise to precipitation. Okay, when antigen and antibody present individually in the solution, they remain soluble. They do not going to get precipitate. But when they come together, they form a lattice, and now this lattice precipitate out of the solution and become visible. Okay, so we can define precipitation. Okay, so we can make the definition of precipitation like when soluble antigen combine with its antibody, okay, in presence of electrolytes, okay, electrolytes like NaCl. For this reaction, precipitation reaction to occur in the solution, antigen and antibody require electrolytes, and well known is NaCl, sodium chloride. Okay, so when soluble antigen combine with its antibody in presence of electrolyte like NaCl at optimum or suitable temperature and pH, a visible precipitate produce. Okay, precipitate produce, and it is called as precipitation reaction. Okay, so we can make final definition like when soluble antigen combined with its antibody in presence of electrolyte like NaCl and at optimum or suitable temperature and pH, because this precipitation require optimum temperature and pH. At high temperature, the antigen or antibody molecule get denatured because they are proteins. So this reaction require suitable temperature and pH. Okay, because pH also play important role in maintaining structure of the antibody constant. If the pH varies, the protein get denatured or the overall 3D structure of the antibody get disturbed, and due to which the precipitation reaction could not occur. So when this soluble antigen and antibody in presence of electrolyte combine together at optimum temperature and pH, they produce visible complex or lattice, and it is called as precipitate, and the reaction is called precipitation. Okay, so this is all about the introduction of precipitation. Now there are some conditions which is required for precipitation reaction. Okay, so conditions which require for successful precipitation reaction. Number one is antibody must be bivalent. Okay, the antibody must have two antigen binding site. Okay, first criteria. Now the next condition is antigen must be bivalent or multivalent. Bi or multivalent. The third condition is the antigen and antibody should be present at optimum concentration. Okay, so antigen and antibody must be present at equal concentration in solution. Okay, so the concentration of antigen and antibody must be equal in the solution that ultimately produce precipitation reaction. The next condition required for precipitation reaction is electrolytes, presence of electrolytes, then optimum temperature and pH. Okay, and this precipitation reaction can occur in liquid medium as well as in gel medium. Okay, so remember that thing, the precipitation reaction of the antigen and antibody can occur in the liquid medium as well as in gel medium. This gel may be produced with the help of agar, agarose or polyacrylamide. Okay, so all this type of gel can be used for precipitation reaction. When this precipitation reaction occur in the gel, it is called as diffusion. Why we call it as a diffusion when it occur in the gel? Because in the gel, antigen or antibody diffuse towards the other molecule, so it is called as diffusion reaction. Okay, one can study the precipitation reaction well with the help of precipitation curve. What is precipitation curve? It is the graphical representation of this precipitation reaction. Okay, it is the graphical representation of precipitation reaction. Okay, to understand the precipitation curve, you must first study the reaction or the condition where this precipitation curve can be suitable, okay, or can be made. Okay, in the precipitation curve, we generally use a graph with x and y axis, and we take two different parameters on x and y axis. On x axis, we take increasing concentration of antigen or antibody. Here I am taking the concentration of, say, antigen. And on the y-axis, we take amount of precipitation, amount of precipitant. Okay, what is amount of precipitant? If for the precipitation curve, I take 6 or say 5 test tubes. And now, in each test tube, I added Okay, now in all 5 test tube, I added equal quantity of antibody or antiserum. Okay, now in all tube, I add increasing concentration of antigen. Okay, so now I add antigen in increasing concentration. And in this tube, the precipitation reaction will going to occur. A lattice is produced, a precipitation complex is produced, and now you can sediment out or you can precipitate out the complex with the help of centrifugation process. Now you have to do the centrifugation of these all tubes, and now finally by discarding the supernatant you can measure the quantity of precipitate. Okay, and that quantity which produced in all the tubes is take on the y-axis. So on the y-axis, we take amount of precipitant produced in each tube, for example, okay? And on the x-axis, I have taken concentration of antigen because I had increasing concentration of antigen in every tube. We do not consider antibody for precipitation curve because the concentration of antibody is constant in all tubes. Okay, so we just have to take concentration of antigen. For example, in first tube, I add 2 microgram per ml of the antigen. In the second tube, I add 4 microgram. In the third tube, I add 8 microgram. Fourth, 16, and in the fifth, 32 micrograms. Okay, so in the first tube, 2, 4, 8, 16, and 32. These are the concentration of the antigen. So I have to take this concentration on x-axis. 2, 4, 8, 16, and 32 micrograms per ml, for example. Now, when I plot the graph of antigen concentration versus amount of precipitant produced in every tube, I'm going to get bell-shaped curve. Okay. Now, initially you can see the precipitant produced in the tubes are very low. Okay. When we increase the antigen concentration, the reaction will going to start and the speed of the reaction increases. So we're going to get more and more precipitant. Okay. The peak is reached at, for example, 8 microgram per ml antigen concentration, and after that, if we increase further the antigen in the test tube, no longer the reaction will going to proceed. The speed of the reaction again decrease, and finally it becomes 0. Okay, so this is the bell-shaped curve, which is called as precipitation curve, and it obtained when we increase the concentration of antigen in the tube. Now we can divide this graph into 3 parts. Number 1 is called as Prezone. Number 2 is Postzone, zone of equivalent zone. And number 3 is Postzone. So we can divide the precipitation curve into pre-zone, equivalent zone, and post-zone. What exactly happened in these 3 zones? In case of pre-zone, the reaction is very slow. The amount of precipitant produced is very less, and this has happened because in this zone, antibody concentration is fixed, but antigen concentration is very low. Okay, because it is 2 or 4 microgram per ml, and due to this, many antibodies like the Y-shaped structures remain free in the test tube. Only few antibody can get attached with the antigen. So this is the antigen. Very less cross-linking occur. Very less cross-linking occur. and due to the free antibodies, no lattice formation occur. Lattice formation is important step in the precipitation reaction. The complex remain very small and it couldn't get precipitate out from the solution. And because of this, at very low antigen concentration, like tube number 1 or 2, that is 2 or 4 microgram, in this 2 tube, we're going to get very less precipitation. And this type of reaction or zone is called as Prezone. Why we call it as a Prezone? Because it occurs before the actual precipitation zone or zone of equivalence. Now, in the next zone of the precipitation curve, which is a zone of equivalence, the amount of the antigen and antibody is nearly equal in the tube, like in tube number 8. Okay, the antigen and antibody concentration become optimum, so that all the antibodies or maximum antibodies get connected with one another with the help of, say, this is antigens. So bivalent antigen connect all the antibodies to one another that produce large immune complex, which is a lattice, that precipitate out. So in this tube number 3, we're going to get highest amount of precipitation reaction. So this curve reach to the peak at second zone. Now, if we further increase the concentration of antigen in the tube number 4 and 5, okay, then what will going to happen? Now the post zone come. In the post zone, the concentration of antigen is more than the antibody, and in this zone, what will going to happen? The antibody will going to remain free. They unable to get cross link because as antigen are in excess, each antibody will going to bind to different antigens. So cross linking do not occur or occur at a very low rate, like this. Few antibodies get connected with one another, and due to which many antigen also remain free because antigen are in excess in the solution. So this complex unable to get settled down or precipitate. So we're going to get low amount of precipitant and the reaction slow down in the post zone. So these are the 3 important zones of precipitation curve. Number 1 is Prezone. In this case, antibody is in excess. Antibody is in excess. And antigen is in low concentration. In case of equivalent zone, both are equal or both have optimum concentration. So we call it as an equivalent zone. And in case of post zone, antigen are in excess. Okay, so with the help of this diagram, you can study the precipitation reaction well. Okay, so you have to understand this diagram before starting the study of precipitation reaction. Now, there are various types of the precipitation reaction. And in this series, I am going to discuss 3 important types, like number 1, precipitation ring test. This precipitation ring test is the type of reaction which occur in the liquid medium. Okay. Then second type is single radial immunodiffusion. This single radial immunodiffusion is also called as SRID test. Okay, single radial immunodiffusion, or it is also called as radial immunodiffusion, that is RID. Another name of this technique is Mancini test. Okay, single radial immunodiffusion is also called as Mancini test. The third type of precipitation reaction is double immune diffusion. And this double immunodiffusion is also called as Auchterlonie test. Okay, so single radial diffusion is Mancini test and double immunodiffusion is Auchterlonie test. The last type of the precipitation reaction is immunoelectrophoresis. So in this video series, I am going to discuss all these type of test which is based upon precipitation reaction. Thank you.